Bismillahirrahmanirrahim. So today we are going to talk about epigenetic cell memory during development. So this whole concept is very fascinating. This is basically one of the most cutting edge topic in molecular genetics. It is linked to basically the epigenetic phenomenon which plays a major role in our development. For example, why eye cells, which are determined early during development, they remain eye throughout our life. Liver, kidney, heart, muscle cells, etc. Their fates, they are determined very early during development, like within first 3 to 4 weeks of our development when we are in our mother's wombs. But once those cells attain their specialized or specified cell fates, these fates are maintained throughout the development, and this is basically due to cell type specific gene expression patterns which are established there. However, if we are to understand epigenetic cell memory, we have to understand the relationship between genes and chromosomes. We have to understand what is a gene. Because if you understand what is a gene, it will be very easy for you to understand then what is epigenetics. If you are able to understand the relationship between genes and chromosomes, you will be able to understand very easily epigenetics. So I have divided this topic into different sections in such a way that first we are going to cover what is the relationship between genes and chromosomes, so that we have a clear understanding what is gene, and from there we'll talk about how we understand the inheritance of genes. So if we understand in this section what is a gene, how genes are related to chromosomes, and the chromosomes eventually get inherited, which is the fundamental genetics, it will be very easy for us to then understand what is epigenetics and eventually the epigenetic cell memory. So what we know that in a eukaryotic cell, let me draw a eukaryotic cell. So in a eukaryotic cell, you have genetic information. It exists in the form of chromosomes. So these thread-like structures I have drawn within the nucleus of the cell, these are representing, 2 pairs of chromosomes in a eukaryotic cell. Now, if I just take one chromosome and draw it for your understanding, so let's say we take one chromosome. So chromosome looks like this. And chromosome is basically this, what we call the thread-like structure, which harbors all the genes. It's made up of 2 components. There is a DNA molecule, double helix, and this structure of chromosome, here I have just drawn a linear thing. It's not linear like this. The chromosome is made up of, a molecule of DNA plus chromosomal proteins, which we call histones. So if I now take just a tiny part of this chromosome, just very little part of chromosome, and I try to zoom in and go inside what actually chromosome is made up of. So as I said, a chromosome is made up of specialized proteins, the chromosomal proteins, which are integral part of chromosomes, and these proteins are called histones, histone proteins. Normally what histones do, they have like H2A, H2B, H3, H4, so 2 molecules of each. So the chromosomal histones are H2A, H2B, H3, and H4. H3 and H4, they make tetramer, and two dimers of H2A, H2B, together it makes a histone octamer, 8 molecules. Histone octamer. I repeat again, 2 molecules of H2A, 2 of H2B, H3, H4, 2 each. It makes 8. H2A, H2B, there are 2 heterodimers of H2A, H2B, and then there is a tetramer of H3, H4. Now, this histone octamer, it contains DNA wrapped around it in 2 circles like this, and then you have the next histone octamer. Again, DNA wraps around this histone octamer in 2 circles, and then the next histone octamer. H2A, H2B, H3, H4, and so on. And DNA, so on, it goes like this. And this is how this thread-like structure is formed. And you know, these DNA packaged in this manner, it undergoes many folds, you know, many higher order structures before it becomes visible to us in the form of chromosome. It's never a linear structure. Remember, there are many loops and, you know, 30 nanometer fiber, you know, the solenoid structure, etc. So this one unit, this one, is called a nucleosome. Nucleosome is 1 histone octamer plus 146 base pairs of DNA. We call this a nucleosome. In a eukaryotic cell, if there are 6 billion base pairs in a diploid cell in humans, so 2n, 6 billion base pairs in a human cell, single cell, okay? And if each nucleosome wraps around 146 base pairs, you can imagine how many nucleosomes are there. It will not be precise 6 billion divided by 146 because it depends, you know, on the chromosome structure, you know, loosely packed and tightly packed. I'm referring to heterochromatin and euchromatin here. So it's very important for us to understand this packaging part of DNA or the genome within the nucleus. This is fundamental to understanding the epigenetics and then subsequently the epigenetic cell memory. Now, this fundamental unit of chromosomes, which I refer to as nucleosome, many nucleosomes together, we call it chromatin. For example, this is, you know, a piece of chromatin. This was a chromosome. We took a tiny part, we zoomed in on a specific, you know, sequences of DNA or region of chromosome, and we learned that, you know, chromosomes are made up of nucleosomes, and nucleosome is basically made up of histone proteins. histone octamer plus 146 base pairs, it makes one nucleosome. Many nucleosomes together make chromatin. And now if we go into the two different components of this chromatin to further understand what is actually a gene. Because I said, first I'm going to explain you what is the relationship between genes and chromosomes, and we are understanding the structure, the packaging of DNA, because packaging of DNA, and now we are going to understand the gene. Now, what is a gene? If we now take up this DNA sequence, this blue one, okay, I just write down the sequence of a gene. Normally, what we do, we draw a horizontal, line like this, and we explain it, like this. This is like a piece of DNA which is on these nucleosomes. These hash lines means, this is in the chromosomal context. There are many, sequences present, continuous sequence. Now here, when we define gene, we say, okay, it has specific DNA sequences which we call promoter, where the RNA pol2 or the polymerases bind. Then the gene has the gene body, which contains introns, exons, etc. in eukaryotes, and this region, gene body, and is followed by transcription termination sequences, where RNA pol2, when it arrives here, it falls off. I'm not going to go into details of central dogma, but I just repeat, the promoter elements are, it contains specific sequences like Tata boxes, etc., which are the recognition sites for, general transcription factors, Tata binding protein, TF2D, etc. And we have, this is, promoter element is recognized by the RNA polymerase machinery. It slides through, once it recognizes, then it initiates the transcription, and it slides through the gene body, and you have synthesis of messenger RNA. After synthesis of messenger RNA is complete, it undergoes, of course, it undergoes RNA splicing, the processing of RNA, and eventually you have a protein. We are trying to take, the sequences. We have zipped apart this nucleosomal DNA just to simplify what is a gene. Now, remember, this picture shows that this particular gene is a protein coding gene. It's encoding a protein which performs some function. But remember, not every DNA sequence in our genome is a protein coding sequence. And I give you an example. Not every gene looks like, Tata box followed by, introns, exons, messenger RNA, and then a protein. There are genes which are called non-coding RNAs, non-coding genes as well. For example, I draw it here. So there will be regions within our genome which when gets decoded in the form of RNA. So let's say an RNA is made out of them. This RNA does not get translated. It makes, secondary structures. Such RNAs, they make secondary structures like this. But these RNAs, they don't get translated into protein. Not every non-coding RNA makes such strong structures, because RNA is a single-stranded molecule. It can, due to self-complementarity, it makes such stem loop structures as well. But I give you examples of non-coding genes which you know already. And the non-coding genes are, for example, you have RNA molecules which contribute to ribosomes. Okay, ribosomal RNA, it's an integral part of ribosomal or the translational machinery. You have tRNAs, you have SNO RNA, small nucleolar RNA, you have splicing machinery. The spliceosome contains many non-coding RNA. So the definition of gene, or our understanding of gene, is basically a sequence of DNA which gets decoded. The information in that particular gene is decoded, and it plays a certain functional role in a cell. It contributes to a specific cellular process. For example, here I talked about translation. I talked about splicing. These genes exist, but these genes, they do not get translated in the form of a protein. The hidden code in these genes is just decoded, and they remain as a non-coding RNA. Last 20 years have also discovered that in addition to this ribosomal RNA, splicing or spliceosomal RNA, we have other forms of non-coding RNA which are called siRNAs, short interfering RNAs, microRNAs. siRNAs, they contribute to post-transcriptional level of interference in transcription. MicroRNAs, they were initially discovered as non-coding RNAs which block translation. So starting from chromosome, we have gone all the way how DNA is packaged in the form of nucleosomes, and what actually constitutes a gene. So a gene is basically information encoded in DNA, which when gets decoded in the form of RNA, it gets translated in the form of a protein. So we have protein coding genes, and we have non-coding genes which do not code any protein, but their RNA is very crucial for cellular functions. In protein coding genes, we know they are transcribed by RNA Pol2, and the non-coding genes, ribosomal RNA, tRNA, etc., they are transcribed by RNA Pol1 and Pol3, etc. So I repeat everything. So the relationship between what we covered is what is a gene? It's the specific region of a DNA which is packaged with nucleosomes, which makes part of a chromosome, a tiny part of chromosome here, maybe just this position, and it contains certain information, certain coding information, and they can encode proteins. They will be protein coding genes, and they are non-coding genes. If I draw, for example, just a piece of chromosome, we may have these rectangles are, for example, protein coding genes. And these regions intergenic region, these are the places in the intergenic region. Normally you have these non-coding RNAs coming up, and these non-coding RNAs can be, long non-coding RNA, like we have in the imprinting genes, in the X chromosome inactivation, they can be, these non-coding RNAs can be siRNAs, microRNAs, etc. So what in this section we have learned is that genes are specific regions of DNA packaged in nucleosomal form, and the information encoded by DNA is decoded in the form of messenger RNA or non-coding RNA, which can be either translated in the form of protein if it is a protein coding messenger RNA, protein coding gene, or non-coding RNA can be in the form of, coming from ribosomal DNA, which a ribosomal RNA contributes to the translation in being part of ribosomes, or small nuclear RNAs which are part of splicing machinery, or microRNAs or siRNAs, etc. There are different kinds of non-coding RNA. So in the next part, we are going to now understand the relationship between genetics and epigenetics, and then we will learn how epigenetics plays a role in our development. So stay tuned and watch the next part.